Welcome, everyone. John Bickle is a very well-known philosopher of neuroscience, graduated in California at Irvine, then got a professorship at Cincinnati, where he also was head of the department, and then he became an adjunct professor here at Mississippi State University, where he's also affiliated faculty department of neurobiology and anatomical science at the University of Mississippi Medical Center. And as you know, John is a very prolific philosopher, working and writing on various themes in the philosophy of science, mind, and neuroscience. And his further work on scientific reductionism or the impact of logical positivism in philosophy has been quite influential in philosophers. And he's known for his ruthless reductionism, the main advocate of cellular and molecular cognition in neuroscience, for his rigorous attempts to link mental phenomena to molecular mechanism. And recently he's also engaged in new metascientific projects on the practice and significance of tool experimentation in the history of neuroscience and in contemporary neuroscience. Just a couple of words on how John is prolific. He wrote more than 90 articles, 3 books, and a very helpful handbook of philosophy of neuroscience, and the 2 important entries in the Stanford Encyclopedia of Philosophy that I'm sure many of you already benefited of. I would like to give you the stage, John. So, John, the stage is yours.
Well, thanks, Antonella, and thanks again. One of the things I've really been lucky in throughout my career is to have the opportunity to work with truly excellent graduate students, postdocs, and this continues on to the present day. As many of you know, Antonella is postdocing with me this semester, and it's just been, and there's many other people in the audience who's worked with me here, and it's just been great. I've had the opportunity to interact with so many smart people with so many different views, who were receptive to listening to me talk on and on and on about molecular neuroscience. Look, today I want to do something that's in line with a lot of my previous work. But instead of going after cognitive and systems neuroscientists for not paying enough attention to the cellular and molecular details, I'm going to go after even a bigger game today. I want to talk about some limits on computational modeling from within mechanistic neuroscience. And as Alok pointed out earlier, let me be clear that by mechanistic here, I'm not specifically talking about the new mechanism. I'm using mechanistic in the title much more broadly here. The approach in neuroscience, especially neurobiology, to discover mechanisms of neurobiological phenomena. Now, one of the things I want to start is by calling your attention to just how prominent quantitative modeling, which nowadays goes under the heading of computational modeling, has become throughout neuroscience. Now, this is nothing new. Neuroscientists have been using computational or quantitative modeling for decades, but it's really become more and more prominent as we've gone along. And as this nice little cartoon kind of demonstrates, the computational modelists think of themselves as sort of the hip and trendy, people in this discipline versus the stodgy old wetlabbers, as this cartoon kind of nicely illustrates. And I just want to make sure everybody knows that this impact of computationalism is intended to stretch into the search for mechanisms of brain function. Now, a really eye-opening example of this is from a recent survey by some computational neuroscientists, where these particular people surveyed 113 different authors of computational neuroscientific papers and asked each of those authors to rate the goals of up to 3 of their authors' own publications, according to a list of 12 goals that the surveyors derived from numerous landmark publications in the field. And the most popular goal overall of the survey among all of these computational neuroscientists was the one that was labeled interpretability. And here was the phrasing for this goal. Some models can be easily interpreted with respect to how they work, e.g. what outcome they predict, and or how the brain might implement the computation, e.g. the underlying causal mechanisms. That was the most popular goal chosen by the 113 people surveyed here. Another goal very near the top of the list, especially under the general category scientific impact, was the goal titled Inspire Experiments, with the phrasing, some models change the way we think about a problem and thereby raise interesting new hypotheses. This can then inspire new experiments. Now, I want you to understand what's being asserted here by the computationalists. They are asserting a primary epistemic role for computational models as guides for wet lab experimentalists. And it's really important to notice that this guiding attitude is held by computationalists about what many computationalists recognize as a landmark publication in the field of computational neuroscience. And that is this 1952 paper by Hodgkin and Huxley, a quantitative description of membrane current and its application to conduction and excitation in nerve. This paper was the 5th of 5 1952 publications for which Hodgkin and Huxley were awarded 1/3 share each of the 1963 Nobel Prize for Physiology or Medicine. Now, this paper, this 1952 D paper, is the paper in which they offer their so-called, what's now come to be called the Hodgkin-Huxley model, a mathematical model of the voltage-dependent, the time-independent properties, the voltage and time-dependent properties of the sodium and the potassium conductances. A coupled set of differential equations describing the ionic basis of the action potential. Now, the thing to point out about this paper has now been cited over 23,000 times in the PubMed literature, and it continues to be cited numerous times to the present day. Interestingly, none of the other 4 papers in the 1952 series has been cited more than 4,000 times and is no longer cited very regularly at all. Now, just to give you the flavor of what computational neuroscientists think about this Hodgkin and Huxley 52D paper, here's a beautiful quote from Michael Hausner, from his much-cited Nature Neuroscience review, where he says, Hodgkin and Huxley's most remarkable achievement was an empirical representation of the experimental data in a quantitative model, the first complete description of the excitability of a single cell. And if you will see similar remarks by people like Christoph Koch and other kind of, very, very prominent computational neuroscientists when they remark explicitly about the Hodgkin and Huxley model. This attitude has also become kind of a common component of all of computational neuroscience, including the kinds of things that philosophers have said about computational neuroscience. So, for example, from the Computational Neuroscience Society's website, you will find there that they note that the Hodgkin-Huxley model is one of the foundations, or is a foundation of, quote, the study of brain function in terms of the information processing properties of the structures that make up the nervous system, whereby, quote, computational models are used to frame hypotheses that can be directly tested by current or future biological or psychological experiments. But I want to point out, and try to make the case today, based on a historical study of the Hodgkin and Huxley work, that this attitude about the assumed direction of influence, computational models directly influencing novel wet lab experimentation, is very much a myth of the computationalists. And I think if we look at the actual scientific achievements and the actual scientific work that Hodgkin and Huxley did that accumulated in their 1952 series of publications, including the first one with Bernard Katz, beautifully illustrate the points I want to make today. So I want to focus today on the historical record of the science that Hodgkin and Huxley actually pursued from 1947 to 1952. I want to look at what they actually published in their 1952 series of papers. And I also want to look at their own published evaluations of their quantitative model. So I'll address each of these points in turn. So let's start with the actual historical record of Hodgkin and Huxley's science from 1947 to 1952. Now, one of the first things to point out is that Hodgkin and Huxley's beautiful computational model, and here I'm just using a quip from Thomas Henry Huxley's famous quip, who was actually Andrew's grandfather, was a result of two ugly, then new experiment tools. The squid giant axon, and I'm of course not talking about axons from giant squids, I'm rather talking about the axon of the stellate nerve that basically innervates the squids and drives the squid's jet propulsion motor system through an aquatic medium. Now, the remarkable thing about this nerve, the giant axon of the squid, is its diameter. It has a diameter in typical squids of 1/2 to 1 millimeter. Squids, being invertebrates, don't have myelin as a way of speeding up the conductance of action potentials down axons. Instead, they've evolved thicker diameter axons to speed up conduction. And this the conduction speed is really important in this particular neuron, in this particular nerve. And so we have a nerve that had these remarkably huge diameters. You're looking on that plate at a photograph of the actual squid giant axon. Now, Hodgkin and Huxley actually started using this tool well before 1947. In fact, back when Huxley was still Hodgkin's student, they published their first paper using the squid giant axon in Nature in 1939. And this paper is entitled Action Potentials Recorded from Inside a Nerve Fiber. They were able to actually make recordings of action potentials and of membrane potentials from inside the nerve fiber because you could actually get the electrodes of the day into the center of this huge diameter nerve. But then, of course, between 1939 and 1947, World War II intervened. Both Hodgkin and Huxley volunteered for British military service and served in the war effort, and I'll just note that those were different times. But after Hodgkin and Huxley reunited after World War II, they had the squid giant axon as an experimental preparation, but they also had access to an even newer tool, the voltage clamp, which had been invented by the American Kenneth Cole in 1947. Hodgkin quickly went to America and came back with the voltage clamp, and Hodgkin actually came up with a modification of the voltage clamp. Cole's voltage clamp only had a single electrode. Hodgkin's turned it into a 2-electrode preparation where a recording electrode is inserted into the squid giant axon. That gives you a continual recording of the action potential from inside the axon, which is fed into a feedback circuit to a stimulating electrode. And so you use a command voltage, and so you can literally clamp the voltage of the potential at any particular membrane potential you want. And then you can experiment by altering ion concentrations in the fluid that are bathing the neuron. This is the tool that Hodgkin and Huxley immediately put to use in early 1948. And what they were able to do was, using this tool and the squid giant axon, directly measure a fast but transient inward sodium current and a delayed extended outward potassium current, and mathematically combining these two currents approximated the measured action potential in the squid giant axon. Now, to the question, which came first, the quantitative model or the experimental data? In Hodgkin and Huxley's case, there's no question whatsoever. The experimental results, 4 years of virtually daily experiments with the squid giant axon and the voltage clamp, preceded the theoretical model. Now, what I want to do now and move on is to my second point, because I think this dynamic of the experimental data driving the quantitative model is beautifully reflected in the 1952 series of publications where they report to the scientific world this 5 years of results. Here are the first 4 papers in this 1952 series. The initial paper by Hodgkin, Huxley, and Katz, and then the 1952 A, B, and C papers by Hodgkin and Huxley. And I want you to take note of the affiliations from the Laboratory of Marine Biological Association, Plymouth, and the Physiological Laboratory, University of Cambridge. I'll come back to that point in a minute. But these were the first 4 publications. And then there was the 5th publication, the quantitative description publication. And I'm going to say something about what was involved in all of these and call your attention to some of the features of these publications. With regard to Hodgkin, Huxley, and Katz, what gets described in this paper is the voltage clamp, the setup and the experimental procedures, and the special two electrode preparation that Hodgkin and Huxley had designed off of Cole's basic voltage clamp. They also report in this paper the first measurements of ionic currents, particularly sodium and potassium currents, after clamping membrane potential to different voltages. And the effects, they report effects on both the sign and the time course of the ionic currents, which varied extensively with changes to both clamped membrane potential and with temperature. In the 1952 Hodgkin-Huxley 1952A paper, we have results reported on altering sodium concentrations in the nutrient bath, on membrane current potential, and evidence for both the fast but transient inward sodium current and the delayed but longer duration outward potassium current. And they were able to show that the total measured membrane current is resolvable into the measured sodium and potassium currents, and that this experimentally established relationship between sodium concentration and current matches a theoretical equation, assuming that sodium and potassium currents are independent of each other. In the 1952B Hodgkin and Huxley paper, they report additional experimental data with independent manipulations of both sodium and potassium currents. They also report additional data suggesting that the rising and the falling of both the sodium and the potassium currents are functions of membrane depolarization and hyperpolarization based on this wealth of experimental results. In the 1952C paper, they report data from additional detailed voltage clamp experiments measuring sodium conductances to even smaller changes, incremental changes in the membrane potential. So these 4 papers collectively report 4 years of wet lab bench work spread out over these 4 publications, which appear in 80+ consecutive printed pages in what was then the world's foremost physiology journal. That brings us to the 1952D paper, the one that gets all the press among computational neuroscientists. Now, I want you to notice something about this publication. First of all, it was published in a different issue of the Journal of Physiology. Same year, 1952, but volume 117, not volume 116, and 500 pages into volume 117, months after the original 4 publications. Also notice that Hodgkin and Huxley for this paper have changed their affiliations. The laboratory of the Marine Biological Association in Plymouth has been dropped. Notice also the paper's title, which is quite revealing of what Hodgkin and Huxley take themselves to be doing. A quantitative description of membrane current and its application to conductions and excitation in nerve. And I also want to call your attention to the title of section 2, where Hodgkin and Huxley first present their quantitative model. The title of that section is Mathematical Description of Membrane Current During a Voltage Clamp. And those are my emphases. Now look, throughout this publication, this 1952 publication, Hodgkin and Huxley are explicit about a couple of things. First, they're absolutely explicit about the model's source in the experimental data reported in the first 4 1952 publications. So here's a rich passage, one of many, from the paper's discussion section. Quote, the results presented here show that the equations described in part 2 of this paper predict with fair accuracy of the electrical properties of the squid giant axon, the form, duration, and amplitude of spike, the conduction velocity, the impedance change during the spike, the refractory period, ionic exchanges. This is a satisfactory degree of agreement since the equations and concepts were derived exclusively from voltage clamp records. That's my emphasis. Now let me go back here to this, because there's another thing that Hodgkin and Huxley are absolutely explicit about throughout this 1952 paper. And this brings up my third point today, that Hodgkin and Huxley published acknowledgments of the limitations of their quantitative model, because they're absolutely explicit about the limitations of their model throughout this paper. Let's go back to this passage from the description section that I was focusing on. Here's how it continues. The agreement must not be taken as evidence that our equations are anything more than an empirical description of the time course of the changes in permeability to sodium and potassium. And they continue a little later on in the same page. Certain features of our equations were capable of physical interpretation. And in fact, Hodgkin and Huxley's physical interpretations led them to some of their choices of equation parameters. But they also remark explicitly that, quote, the success of the equations is not evidence in favor of the mechanisms of permeability change that we tentatively had in mind when formulating them. I mean, the quantitative model couldn't provide evidence for that, and being developed back in Cambridge after 4 years of wet lab experimentation, their quantitative model certainly didn't inspire those experiments. Now, I want to point out that by the time Hodgkin and Huxley were awarded their Nobel Prize, December of 1963, they were under no illusions about whether their quantitative model would help in the search for molecular mechanisms of the action potential. Now, by this time, their quantitative model was being quite famous, but they were under no illusions. And I want to call your attention to a couple of the passages in their Nobel Prize addresses that point this out. First, this is from Hodgkin's Nobel Prize address, 1963. Quote, to begin with, we hope that the quantitative analysis might lead to a definite molecular model of the membrane. However, it gradually became clear that different mechanisms could lead to similar equations and that no real progress at the molecular level could be made until much more was known about the chemistry and fine structure of the membrane. And Huxley, in his Nobel Prize address, is a bit more favorable about the quantitative model. In fact, he describes the model in his address, but he closes with the following point. Both Hodgkin and I feel that these equations should be regarded as a first approximation, which needs to be refined and extended in many ways in the search for the actual mechanisms of the permeability changes on the molecular scale. So basically, both are urging is, if we're going to make progress on the molecular mechanisms of the action potential, it's back to the lab bench. Now, it was another 2 decades before the development of another new tool, before the ion channels and the action potentials we know and love today were first characterized. But these revolutionary discoveries were guided by no quantitative model. They were directly the result from a then new experiment tool developed in the mid-1970s, early into the 1980s. That's Naher and Sackman's patch clamp, which enabled experimenters to measure the picoampere currents through individual ion channels and then excise those channels and subject them to the X-ray crystallography that revealed their protein structure. So like all revolutions in mechanistic neurobiology, these molecular mechanisms of the action potentials were, if not turtles all the way down, certainly wet lab tools and experiments all the way back. So I'm going to insist that Hodgkin and Huxley's point about their famous quantitative model apply to computational modeling efforts today. These models are descriptive of already gathered experimental data or phenomena. They are epistemically dependent on the experimental data for all their empirical veracity, not vice versa, and they are at best of heuristic use in guiding experimental work aimed at discovering mechanisms of neurobiological functions. And historically, at least, in wet lab neurobiology, they have been of very limited impact, even in this fashion, with the place and impact of Hodgkin and Huxley's famous model being just one example from many that I think we could talk about. So all of this, I think all of this is true for the very reasons that Hodgkin and Huxley noted 60 years ago. Different mechanisms could lead to similar equations, and computational models, especially those implemented on digital computers, are at best approximations of actual neurobiological processes. These are logical points about computational models. They thereby can't determine mechanisms. So the take-home lesson today is this. If it's neurobiological causal mechanisms that you seek, and I'll admit there can be other projects in neuroscience, but if you're after neurobiological causal mechanisms, if that's what you're going after, there's nothing else to do but back to the wet lab. And quit effing modeling. Thanks. I will quit here and take on what I know will be many questions and many challenges. And I'll take my inspiration from Bobby Fischer here, who at age 21 took on 50 opponents simultaneously in 1964, winning 47, drawing 2, and losing 1. Thanks.
Thanks a lot, John. Okay, Alok, please.
Yeah, John, very exciting talk. I'll give a little 2-sentence background to where my excitement is coming from. As an undergrad in 1985, I got to go to my first molecular biology lab, somebody who had done a late postdoc with Seymour Benzer, who you might know had isolated the Shaker mutant in Drosophila. And I got to learn to do electrophysiology recordings in the proboscis of the Drosophila and actually get Bernard, the cats and Huxley papers and worry about, I didn't do any patch clamp. 5 years later, I got to present for Journal Club, the paper by Dick Aldrich on the ball and chain model of the Shaker channel. Right, and then how it closes in. And and subsequently I finished my PhD in molecular biology, realizing that whole things have different behaviors than the molecular mechanisms or and I really appreciate I don't know what to characterize it, you're deconvolving the presumptions of mathematical modeling away from an explicit, almost metaphysical epistemic status to that these are mathematical thumb rules and or heuristics, and their accuracy of characterization doesn't extend down to the depth of the layers of material behavior, right? It reminds me of the situation in early 1800s where you had extremely good mathematical descriptions of the behavior of matter on the hands of Laplace and Young on mechanical twisting and the equations of the surface tension of liquids and the squeezability of materials, you had beautiful equations that you could do engineering with. And you were way before the atomic nature of matter and the particulate description of, it's a pre-Boltzmann, pre-Dalton world. And the mathematical models were completely actionable. That's what I kind of am very excited by your pulling up the history of the constructions in the 5 papers and the scientists themselves saying the equations do not give us wings to escape the lab.
Yeah. Yeah. No, thanks, Alok. I mean,
that's all I wanted to say.
That's an excellent example. But it also, there's another point here too that I think is really worth pointing out. And that's this. I am always really reluctant to try to draw lessons for neurobiology or neuroscience from physics. So because I think that's just I think that the extent to which mathematics and even something like theory are at work in physics and the way that they're at work in neurobiology are just fundamentally different. And so while it could be the case that people can come and say, well, look, we've got all these great things and where these computational models were so important in driving lab work in physics. I said, okay, that may well be the case, but the history of neurobiology, when we look at the landmark discoveries that we teach in an undergraduate introduction to neurobiology class, I can't think of a single one where a computational model was inspiring the experiments that led to those discoveries.
Marco, please.
Thank you, and thank you, John. This was, as usual, interesting, provocative, and illuminating. I have a lot of questions, but for the sake of time, I'm going to confine myself to a general remark and a more substantive question.
Sure.
My general remark is, why do you bring mechanism into the picture? I mean, it seems to me that if I understood your talk correctly, and feel free to correct, of course, it seemed to me that your target, which is a really interesting one, is mathematical modeling. And to me, to reshuffle things and bring mechanism to the picture, on the one hand, it gives mechanism too much due. I happen to think that it already has too much attention in the field of philosophy, but also it kind of muddies the water because then it's not clear at what level mechanism comes into the picture. So why not just target mathematical modeling?
Yeah, no, that's fine. That's one of the things I said at the very beginning. I want to make sure that I'm not here meaning new mechanism in the sense of McNamara, Darden, Craver, or Bechtel, or any of the others who have sort of really brought mechanism to prominence in neuroscience. I mean it here just as the kind of a much more broader category of just the search in neurobiology for just what are characterized scientifically, not in terms of a precise philosophical definition of the search for mechanisms of neurobiological phenomena. Yeah. So I have in mind the kind of work by which we try to find work in intercellular signaling pathways that lead to synaptic plasticity, work in new gene expression, new protein synthesis that restructures synapses in order and tying that experimentally with various animal behavior and learning and memory paradigms. I just have in mind this kind of approach that we might call this kind of the search for mechanisms broadly conceived in neuroscience, especially neurobiology. So I want to make sure that I'm not sort of tempting people to think that this is a broad defense of mechanism. As many of you know, there's a lot I agree with on the new mechanism, but there's a really important point that we disagree about. Me and the new mechanists, particularly the ones that focus on neuroscience, and that's this. And that has to do with their recognition of this nested hierarchy of mechanisms within mechanisms. And my idea that look, there are scientific approaches that are literally trying to find the molecular mechanisms of cognition, not the molecular mechanisms of synaptic plasticity, which are the mechanisms of place cells, which are So the difference, so I have my own fight with the mechanists. We have our own agreements, we have our own disagreements. But here, I think I just simply have in mind the kind of approach, for example, when we see in Hodgkin and Huxley, they use the word mechanism in this broad, vague scientific sense. They tie it to molecular mechanisms, and that's the sense of mechanistic neurobiology that I'm after here. That's the sense in which I think computational modeling is of limited use, historically has been of limited use.
And yeah, I completely agree. And if I can transition to my more substantive point, I happen to agree with you. But for the sake of the argument, if I were to criticize what you were saying from
Sure.
different perspective, I would say, are you assuming some kind of causal fundamentalism, where you're kind of speaking in this assumption that all the substantive causes at the neural level occur at the level of in the individual neurons and network and so forth? So what would you respond to someone who said what the approach you're criticizing brings to the table is a nested hierarchy of causal levels where we're investigating different higher level causes of neural networks, which cannot be reduced to
Good. So that's why I said at the end, I made that kind of cursory remark that well, look, there could be other things you're interested in doing in neuroscience besides just searching for this broad sense of mechanisms of various brain functions. But I mean, if you want to push me on it, I mean, I'm pretty willing to go even further. I mean, I think that at least the bulk of neuroscience that interests me are causal fundamentalists, and they also tend to be, I think, pretty clearly causal reductionists when they can find these molecular pathways and manipulate those pathways and show direct effects in some kind of behavioral paradigm of learning and memory, and then claim to have found a molecular mechanism of declarative memory or hippocampus dependent memory. I take them dead serious in that claim. And actually, I think there's an entire branch of neuroscience that just works under that, as you call it, causal fundamentalist assumption. That's the branch I'm really interested in.
Andrew?
Yeah, thanks, John. Great talk, as always. And I'm not going to be your Bobby Fischer opponent. I'm actually going to say something that probably just supplements what you have in mind. So I've also been thinking quite a bit about the relationship between computational modeling or formal modeling and mechanism. And I think the broader sense of mechanism that you have in mind too. And actually, I gave a talk that sort of reflects the way that I think about it a couple of years ago on the Hodgkin and Huxley model. But it's been a couple of years, so I forgot all the details of the Hodgkin and Huxley model stuff. But the talk kind of used this analogy that I think it has anchored my own thinking about the relationship between these two features of science that I think will be illustrative here and supplement what you have in mind. So the analogy comes from Don Norman's book. For those of you who don't know him, he's a thinker in design. He's also an electrical engineer and a psychologist. And he's giving this example that's supposed to illustrate what bad design looks like. And it's the example of this poorly designed refrigerator, right? So he says there's a control panel on the refrigerator and it has these two dials, each of which is surrounded by the numbers 1 through 7. One of them's labeled refrigerator and the other's labeled freezer. And there's an arrow indicating the direction you're supposed to turn the dials to regulate the temperature of the corresponding compartment. And he says, imagine that you're working with this thing and you just can't seem to get it right. And every time you modulate the temperature of the fridge, right, your ice cream melts, and every time you modulate the temperature of the freezer, your milk freezes. And so you're messing with it, and then eventually it dawns on you, like, wait a second, like, I've been understanding this system such that my assumption is that each dial is mechanically rigged up independently to the corresponding compartment, enabling this sort of independent manipulation of the temperature of each. And then it occurs to you, like, wait, maybe one dial is actually regulating the total amount of cold air that gets pumped into the system, while the other one, controls where that cold air gets distributed. And so there's this sort of moment of insight where you realize, like, oh, wait, maybe the underlying mechanics of this thing aren't the way that the outward appearance of it suggests that they are. And I remember thinking when I was reading through the papers that you discussed in your talk, there was a moment in some of the history and the secondary literature that there was a comparable moment in Hodgkin and Huxley where they realized, like, oh, wait, what if the thing works like this? And then they had this sketchy hypothesis, right, like the physical interpretation, as they call it, that they then sort of explicated mathematically, right? But it was the mechanical, physical interpretation that they came to first. That was the insight. And then the mathematical stuff was more of like an afterthought. And it's like mathematically explicating what was first and foremost a mechanical insight. And so I think that illustrates something about the relationship between the quantitative model and the mechanics. And it's in line with what you were saying, because it's mechanism first, afterward, you do the mathematics.
Yeah, no, that's a really interesting point. And here's the way I think it can go. Look, Hodgkin and Huxley did have a mechanism in mind when they were choosing some of those parameters. They had in mind, and they had in mind what they were then calling like voltage gates. And they noted that these voltage gates, by being of different diameters and with different charges, lining the entrance portion of the gate, could explain the independent flow of sodium into the cell and potassium out of the cell, because of the different atomic sizes of those particular ions. They had that mechanistic picture in mind. Now, part of it turned out to be roughly correct when we actually learned what these things are. But there was another aspect that their wealth of data showed that they didn't have any clue as to what that mechanism could be. And that was the voltage dependence of these gates, these separate gates for sodium and potassium, and why the sodium gates were closing down when the membrane potential reached about plus 20, and why the potassium gates were remaining open. They had for the measure, when they found that the voltage dependency of these currents. And even with this gate metaphor in mind, this kind of mechanism metaphor in mind, they didn't have a clue as to what could account for that voltage dependency. And in fact, we didn't have a clue even after the patch clamp, right? Even after the patch clamp in those early works in the 80s, it was another 15 years until we basically got those wings, those charged wings on the proteins of those ion channels that had positively charged amino acids on them that enabled those wings to bob up and down in the membrane with the changing membrane potential to open and close those gates. So I mean, there were aspects of the mechanistic idea that they had that certainly sort of influenced their decisions about some of the parameters of their computational model. You're exactly correct there, and that's an important point. But there were also just flat out limits on their mechanistic imagination as to what in the world could be providing for the voltage dependency of these two, of whatever the mechanisms of these two currents were. And that was only found by, another 40 years of patch clamping and X-ray crystallography and you name it.
John Park?
Yeah.
Hi, John. Thanks for your talk. I was wondering, how much do you think maybe the reason why these mechanistic models, these models, mathematical models, lack direction in or lack influence in these kinds of neuroscience experiments, how much could it be due to the perhaps early stage of neuroscience itself in gathering phenomena to initially describe? So maybe science is still in its early fields, and of course mathematical and theoretical models are going to have to first describe the phenomena first, but then when you have more complex models, they actually start making more novel predictions, and then maybe in the future these mathematical models actually can play a larger role in conducting neuroscience experiments, and there can be a feedback loop where the models get some instruction from the phenomena and new neuroscience tools that we have as far as mapping the brain, etc. But also it helps contribute. The mathematical models can help contribute to the experimentations.
Great, thanks. Maybe. But again, I just kind of look at the history of the landmark discoveries, and I just don't see that particular impact. Now, so I was around when I was around Irvine in the mid '80s. That was the time when so that was a time when Lynch and Gary Lynch and some of the other post synaptic membrane people for LTP were fighting it out with Candel and the presynaptic people in terms of the mechanisms of LTP. And so Lynch had already done his groundbreaking work of showing that calcium in the postsynaptic cell has an effect on whether or not LTP is induced in the synapse. He had already had shown his groundbreaking work on showing that there actually are structural changes at the electron microscope level in the postsynaptic cell. And so next we were already starting. I mean, Colin Ridge had published his paper on the first attempt to characterize the N-methyl D-aspartate receptor and set it aside and show its properties different from what became known as the AMPA receptor. All that stuff was just kind of fresh in the air, right? And Lynch took on a computer scientist, Rick Granger, to help him build increasingly biologically realistic models of the postsynaptic membrane and the postsynaptic environment. So they could then use this model to guide experiments. And so they published papers on this model. It was by far the most neurobiologically detailed model in the mid 1980s of a computational model that for LTP, particularly at the postsynaptic level, at the postsynaptic side. But in the long run, it still had almost no effect on the continual search for postsynaptic mechanisms. I mean, it had no effect until what really drove and what really settled that issue. I mean, it turns out that both Candel and Lynch and others were right. There's both pre and postsynaptic mechanisms. But what really clinched the postsynaptic side had to await into the early 90s and the new experimental tool of gene targeting, where you could manipulate the gene for one particular protein in these intercellular signaling pathways and show the effects on both LTP and in learning and memory in the mutant animals. So again, I mean, I just look at the history of neurobiology and I look at the landmark results in the field, and I can't find one where computational modeling had anything more than just kind of a limited heuristic role in the work that led to those discoveries. Instead, what you find in case after case after case is the ingenious use of a new lab tool.
Greg?
Thanks. What was Kristen on the list before me, or does it not matter?
I'm sorry if I think I've seen your hand before. I don't know, but we have time. Don't worry.
Okay.
Yeah.
Thanks. Thanks, John. So I mean, what I've been thinking about since you finished kind of overlaps with what Andrew and John said. So I'll try to ask it in a different way, or comment in a different way. So one thing, the quantitative model of the action potential is a model of a biological process. So when you're looking at the historical case, I'm not confused about what you're doing, but of course, the quantitative model is going to be based on experimental results. I wouldn't expect it to be different. So the way that the whole Hodgkin and Huxley event unfolded seems to me to be pretty natural and not to really be a problem for the quantitative modeler's position that there's some value to these models. You've just shown that it's based on biological evidence, which is what we want. But so fast forwarding, the example that you were just giving to John's question seems to me like the more interesting one, where, okay, we're no longer just at the beginning of this, but you have people doing models, you have people doing biological work, and they just don't match up in the way that you might think. That seems to me to be a stronger point for your thesis. I don't know what you would say to that. If I could just add, I mean, in other places you've been, dismissive of imaging work or explicating brain processes, but is this just another similarly, are you just as dismissive of computational modeling as you are of, say, fMRI is really missing what the causal process is?
Yeah. Okay. Yeah. Look. So I think about all the lab meetings I've sat in on in molecular neuroscientist labs that I've collaborated with, and I think about what kinds of papers they read from the literature that they're interested in. And I think about the kinds of things that philosophers comment on when they're commenting on neuroscience. And there's just a disconnect. I mean, you just when you hang around these labs, they're just not reading the kind of work, the kind of computational or quantitative or modeling work that so many of the philosophers seem to think defines contemporary neuroscience. I mean, it's not like they dismiss it. It's not like they think it's bad science or anything like that, but it's just not relevant for what they're doing. Whether or not the brain is a Bayesian machine does not matter to someone who's trying to find, like my current collaborator is, trying to find the right mix of genetic manipulations that will turn structure cells in the inner ear into active responding hair cells in the adult rodent model. I mean, but look, that is contemporary neuroscience. That is the bulk of people who are given posters at Society for Neuroscience meetings. The people doing the kind of work that the philosophers are concentrating on and sort of things define neuroscience are basically in one corner of that poster room. The rest of it is people doing this kind of work. This is neuroscience circa 2020. And it just seems weird to me that philosophers of neuroscience would not recognize the fact of what characterizes the discipline. So am I dismissive of fMRI work? Am I dismissive of work in computational modeling for certain purposes? Well, not necessarily, but I just want people to realize how not peripheral, but how small is that community within the broad field of neuroscience, and how much more prominent is the kind of work that I tend to focus on. I think that's an important point if we're going to have a philosophy of neuroscience that's at all descriptive of contemporary neuroscientific practice.
Okay, thanks. Can I just add? Well,
Yeah.
so just to repeat what I said.
Yeah.
I mean, for my part, and maybe other people will agree or disagree, the example you were talking about, where John, about with the pre and post synaptic processes, and then just what you just said there, and kind of society and neuroscience stuff, to me, that seems to make your point somewhat better than the historical case.
Oh, okay, very good. No, but that's why I made the case. You know, that's the way I usually make my argument. But this historical case struck me as particularly interesting because this Hodgkin and Huxley model is still the case that so many computational neuroscientists point back to as the foundation of what they do nowadays, of what they as one of the true success stories of computational neuroscience. You find that in prominent ones. You find it in the Computational Neuroscience Society's website. I mean, this is one of their successful cases for what they think computational neuroscience can contribute. And then you look at these surveys, like this one that was recently done, and you see that these people think that one of the primary goals of computational neuroscience is to contribute to experimental neurobiology. And there's just a disconnect between listing this case as one of your success stories, because this case provided nothing of the sort that contemporary computational neuroscience, at least some of them, claim that computational neuroscience is going to provide.
Okay, thanks.
Christine, go ahead. And sorry if I skipped you before.
No, I don't think you did. I think Greg was on the list before me. So John, I want to ask you about the aesthetic words that came up early in your
Okay, cool.
and I was trying. So I guess I wasn't. So there are a couple different questions. One is, I wondered, you quoted the two tools, the squid, giant axon, and the voltage clamp were ugly. And I was wondering if that quote, if you were quoting something, and if the idea was that tools are ugly, or that these particular tools were ugly for some particular reason.
No, good. Can I address that one, Chris?
Yeah.
It's a great point. So there's this famous quip that Andrew Huxley's grandfather, Thomas Huxley, said about science, and it's just the quip goes something like this: the bane of science, a beautiful theory destroyed by an ugly fact.
Oh.
So that was a well-known quip from Thomas Huxley, who was Andrew's grandfather, from the famous Huxley family in British science. And it just seemed like appropriate to say that this beautiful computational model was actually the result not of this pristine reflection on in Plato's heaven, but was the result from doing this grungy lab work at the Marine Biological Association lab for 4 years using the squid giant axon and this voltage clamp machine. So, but the use of those terms was taken straight out of to paraphrase Thomas Huxley's quip from years before.
Okay. Okay. So this got me started thinking, because I was thinking about. I mean, I like the questions that you're asking, and they make sense to me from a point of view of being really interested in mathematics as a younger
Yeah.
kid. Because I felt like, I mean, when I started encountering people were really pressuring me to take physics because they were like, well, that's what the mathematical ideas don't have any meaning unless you can bring them in contact with the world. And when I encountered the same ideas in the physics, I just felt like the center of gravity of whatever it was that was attracting me to math was not in the fact that they could be brought to bear in this way. And I doubted that, and so it seems to me like you're saying, the center of gravity of the experimental work is not the mathematical, models. It's not, you know, there are different centers of gravity. And then there's an interesting question about why they do so often come together and what the use of that coming together could be. But what got me thinking about that, but I still want to go back actually to the beautiful and the ugly, because I was thinking in mathematics, there is really, I mean, the words beautiful and ugly come up a lot, but they're internal to mathematics. And it's not the idea that mathematics is beautiful and everything else is ugly, but that there are beautiful proofs and there are ugly proofs. And there are things that people mean by that. So there are beautiful mathematical results and there are ugly ones. And part of and so I was puzzled by this.
No, good.
Beautiful and ugly crossing the border, you know.
Yeah. No,
I also wondered if in neuroscience, those terms have their own internal application. So if there is if neuroscientists in their practice do talk about things that are beautiful and things that are ugly, but them being kind of internal to neuroscience, or if those words just don't have a place.
No, good. They do, because I mean, you'll see experiments described as beautiful, you know, or experiments described, you know, or even as ugly. I mean, and they're not talking about the kind of aesthetics of it. Could be a quite beautiful experiment that, you know, if you're there in the lab doing it, it might kind of like turn your stomach, you know. Nevertheless, I mean, it could be a beautiful experiment because of its design, because of its execution, because of the particularly ingenious use it made of these two. So yes, those aesthetic terms do occur within experimental neurobiology. Or, you know, or someone will look at it and say, well, you know, I see your results. But that's not, you know, that's a pretty ugly experiment. So yeah. Terms do occur. Those aesthetic terms do occur in neurobiology, and they have to do with just experiment design, with similar kinds of epistemic and non-epistemic values that they have in these other disciplines. So yeah, they're there.
Interesting. Thank you.
Yeah.
Okay, I will give precedence to Neda first, and then go back to Andrew and Alo. So please, Neda, go ahead.
Hi, John. Thank you. Can you hear me okay?
Yeah. Mm-hmm.
Okay, good. All right. Sorry. I got interrupted a bunch, so I don't know if you answered this already, but I'm going to ask it anyway. So my question is really like, I want to get a better sense of who your target is, because, you know, like a lot of the people I know who do computational modeling, they're doing it to understand something like behavior, right? And so that's, and they don't see it as like an either or where one precedes the other. It's like a back and forth between modeling, going to the physiology, modeling, and going back and forth. So like when I think of something like, you know, people using, thinking about like reinforcement learning with the basal ganglia or something like that, right? Like you're not saying that that's like, you're not being a literalist about it in many cases, right? You're using it to inform you about what the relevant like mechanisms are, what the relevant units are. I'm thinking also of, you know, like people looking at the geometrical structure of neural activity to, again, see what population of neurons are relevant for generating a particular behavior. And so the idea here is that like, for what you're sounding, I don't know if you're setting up a straw man or if your like target is different, but it seems to me that like a lot of computational modelers are interested in different sorts of questions than the kind of low-level neuroscience that you're looking at. So it may not be fair to kind of like, I'm very sympathetic to using the Hodgkin Huxley case, because that is like a very like, that's a trope and it's constantly appealed to by these people. But I mean, using that to generalize in this way, I'm just a little bit skeptical.
No, sure, good point. And again, there can be a lot of reasons to do modeling, and a lot of those reasons can be because you're trying to isolate behavioral parameters or things like that, right? I mean, to where those models are not intended to be a piece in the search for some kind of causal mechanistic story of how the behavior is produced. That could well be, and they could be quite useful in that. I mean, I don't really read that literature, but for all I know, it could be. And I think the reinforcement learning is a really good example. But I think that the computationalists and at least I'm trying to take them literally in especially on that survey of the goals of computational neuroscience. And the two of the most popular goals among that 113 computational neuroscience survey were these goals where the language of the goal had something to do with their use in finding mechanisms and their use in inspiring experiments. And then and that's again go back to Greg's point. And then to say these are two of the most recognized goals among computational neuroscientists, and then to kind of couple that with the fact that the Hodgkin Huxley model is so often chosen or so often presented as one of the roaring successes of computational modeling, all that just simply does not fit together. And that's what I'm trying to point out with this particular example. So yeah, there could be plenty of other uses of, and Hodgkin and Huxley said the same thing about their very model. I mean, they were similarly completely in agreement with the limitations of their model as a tool in pushing forward on finding the molecular mechanisms of the action potential, which of course back in 1963 were still a total mystery. And yet, so here's this computational model that they're already gotten famous for, and they're looking back and saying, in retrospect, Hodgkin's saying, yeah, look, we developed this because we thought it would be useful in the search for molecular mechanisms of the action potential, and it wasn't. It led to no progress whatsoever. And then you turn back and see about the source of their model being so clearly in the 4 years of prior detailed lab work that if that's going to be one of contemporary computational neuroscientists' kind of, shining successes, boy, it sure does not show what a lot of them seem to think computational neuroscience is going to provide.
Andrew?
Yeah, thanks. So I wanted to go back to an observation that you made, John, in responding to Greg's question, and then maybe also try to bring into the fold Nada's question about who your target is.
Right.
And I'm going to do it again using a perhaps annoyingly elaborate analogy. So remember when everybody was talking about the Watt governor? And it was like the touchstone for debates between representationalist and dynamical systems people.
Yeah.
And so like every single paper that you'd read in which the Watt governor was discussed, it would always start like this, like paragraph 1, right? Here's a Watt governor. Here are all the parts. Here's how they fit together. Here's how it fits into the steam engine. And here's how it regulates the speed of the steam engine.
Right.
Beginning of second paragraph. Which of the following two frameworks provides us with a more robust philosophical understanding of the Watt governor? The representationalist one or the dynamical systems one? And at that point, I would always be kind of like, well, I already understand what the Watt governor is doing. Like, what does either of these two frameworks add to that? And so then I asked myself, okay, so they seem to think it adds something. So how are they sort of, organized philosophically such that they feel that something else is needed? Right. And I think the answer is that they're both Platonists, right? Like both of them think that understanding the Watt governor consists in grasping the rationally apprehensible ideal form of the Watt governor. They differ over how to specify that rationally apprehensible ideal form. One thinks, representationalism, and the other thinks DST. but like in essence, they have the same sort of Platonist theoretical aesthetic.
Yeah.
So when you mentioned like why is it that all the philosophers are in the corner of the poster room, right. Where all the modelers are? Right. And I think it's because philosophers generally are Platonists. And so they gravitate toward people who have a comparable theoretical aesthetic. And in neuroscience, the people who have that theoretical aesthetic are the modelers. And so that's who your targets are. To go to Netta's question, maybe what you have in mind is the Platonists, like that's who you're targeting?
Well, look, I mean, yeah, maybe, but look, I think your point is really well taken one. I mean, but here's why I think that philosophers are so attracted to computational modeling. It's just an easier move for people with philosophical training to understand computational modeling than it is to understand how to go and do a gel electrophoresis or how to go in and manipulate a gene to produce another protein, an aberrant protein in an intercellular signaling pathway in a living being. I mean, a living organism. It's an easier fit with philosophical training. The move from kind of what you learn in sort of the basic symbolic logic to computational modeling is just a far easier move than is going and learning the techniques of freaking molecular biology. I mean, so I think that's the reason why philosophers tend to be so attracted to these computational models. They just find them more in keeping with their basic intellectual training. And it is just harder to convince philosophers that sort of neuroscience nowadays is much more doing all this stuff in the lab. It's not so much, develop these computational models. That's so whether it's just kind of in sort of inherent Platonism, or whether it's just kind of the techniques at work within computational neuroscience are just an easier fit for basic philosophical training. Yeah, I think it's something like that. And that's why so many philosophers are focusing on these branches of neuroscience rather than the branches of neuroscience that I try to urge people to focus on.
Alok, do you want to add something?
Yeah, John, I just wanted to resonate with you really. I work on a group of people that do single molecule biophysics.
Mm-hmm.
I'm trying to characterize that community. And they have this interesting situation. They use high-resolution microscopes to hold the molecules. And then they have probes that touch the molecules.
Yeah.
Right? So, but the characterization is sub-wavelengths of light. So what they're touching and what they're pouring into their modeling is not images. And in order to free themselves up to think about mechanisms, the senior people say, we've got to kill the atoms. Okay, and that pushes them back to the era of Thomas Young, Laplace, and good old Rutherford. Rutherford famously was fighting off this Newtonian, the equation is the mechanism. He hated the theory first, right? And so what I sent you an email saying maybe talk.
Sure.
So,
I think it's a great example. It's one I don't know much about, but like, for example, Anne Sophie Barwich and I have started we've collaborated on a bunch of things like this. And she's been really trying to emphasize the way that some of the new microscopic tools that are being used. Now, of course, for her, it's in olfaction neurobiological research, but how these new microscopic tools are so useful for the kinds of work that has pushed the neurobiology of olfaction forward over the past 2 decades, where it's basically brought it out of almost scientific obscurity into mainstream Nobel Prize winning science. So yeah, I think.
What's the name of your collaborator? What is the name of your collaborator?
Anne Sophie Barwich. She's at Indiana. And she's she's been working pretty hard on some of these notions of some of these new microscopic tools and how they're being used, particularly in neurobiology of olfaction. And
Thank you.
Really good, some really interesting ideas on this. I think if the three of us kind of got together and talked, over virtually and talked about some of this, we'd find it a really promising kind of avenue to explore.
Just to encourage that vision, I did work on the molecular neurobiology of olfaction and Drosophila.
Yeah.
So, yeah. Okay.
Yeah, because they can be olfactory conditioned. I mean, Drosophila. I mean, it's fascinating.
Thank you a lot.
Okay, so I have a point that is connected to what we are working on, John, and so maybe it can be useful for a brainstorming here.
Yes.
Definitely. And it is connected to what Neda and Greg were saying before. So I kind of agree with you when you say that philosophers many times or often find themselves more comfortable to reflect or look at computational neuroscience than neurobiological work done in the lab. But they may do another counter argument. And they could say there are certain things that we cannot investigate in human beings. We cannot investigate certain molecular mechanisms and how they work and how they cause intermediate level neural phenomenon that in turn are responsible for behavior. And we philosophers are more interested in explaining human behavior than octopus or mice behavior. And so they could say this is something that maybe we will never really know at that level of analysis and specificity. And so there, could you foresee something like a different kind of integration, collaboration between wetlab work and philosophers and computational neuroscientists?
Yeah. No, good. I mean, because, clearly what we're starting to edge into here, Antonella, is the good old multiple realizability problem. And here's kind of always been my take on this. One of the things that molecular neurobiologists often emphasize when they use an animal model for their interventionist experiments, which of course they have to do most of the time. One of the things they first do is they really do a detailed investigation of the gene and protein that they're manipulating in the animal model and its analog in the human being. And generally what you find, especially when you're working with mice and things like that, especially with the particular genes and proteins that have turned out to be so instrumental, so important for, long-term potentiation and all these other kinds of things, is that you're really dealing with these kinds of housekeeping genes and proteins. And pretty much, at least at the functional regions of both the gene and the protein, a human protein kinase A is a mouse protein kinase A, pretty much is a Drosophila protein kinase A, pretty much is a Aplysia protein kinase A. I mean, there have been residue changes out in the non-functional zones, the non-functional regions, but in the functional regions, they're almost nucleotide base pair to nucleotide base pair match, and they're often amino acid for amino acid matches, almost sometimes identically. And that has always been convincing to me that given these structural and functional similarities of these underlying mechanisms, if we can find those mechanisms in an animal model, and we can find the structural and the functional similarities of those mechanisms in the animal model and the human model, we certainly don't have what Silva and I call convergent 3 evidence in the human. But if we've got convergent 3 evidence for a causal mechanism in the animal model, and if we can show that at least with regard to the intervention we're making, we're intervening on something that is structurally and functionally similar across species, that's enough for me.
Okay, but then you are not explaining the same phenomenon, right?
I think you are. I mean, unless you're a real hardcore, unless you're like Kenizawa and Carl Gillett about realization, I mean, I think you are.
Okay. So, Andrew, do you have another question, or is just your hand from before?
That was just my hand from before. Thanks.
Okay. Then I think we don't have further questions, and I think we can remind all of you that next week we will have another exciting weekly chat with Colin Allen. He will talk of engineered wisdom for learning machine. And thanks for being here and for participating.
Yeah, thanks everybody. Sure appreciate it.
Thank you.
See you, everybody.
Good to see you guys. Bye.
Yeah, see you, Andrew. Great to see you as always.